So our project is devoted to the evolution of the non-triplet genetic code in ciliate Euplotes. First, the genetic code is sort of a set of rules by which the information encoded by sequences of nucleotides is translated into protein sequences. And genetic code is typically like triplet, non-overlapping, and non-ambiguous. For the second two features, there were found multiple exceptions, but the triplet feature remained like one of the most conserved properties in biology in general. That there is such a phenomenon of ribosomal frame shifting when a ribosome changes its frame. It can happen on a specific context or due to some secondary RNA structure or due to some additional factors and so forth. There is a group of ciliate Euplotes where this frame shifting is very abundant and it is 100% deficient. And basically, it doesn't require much more than a short context, basically as stop codon within a reading frame. And it was thus proposed that this feature, it constitutes like a non-triplet addition to the genetic code of Euplotes. And it adds families of 4 and 5 nucleotide long codons due to frame shifting happening on like 1 or 2 nucleotides forward. And so we wanted to see if this change spreads across the genomes of Euplotes, and what are these selective forces that kind of act on these changes. And so we have sequenced transcriptomes of 9 species representing like some of the major clades of Euplotes phylogenetic tree. And we additionally generated a ribosome data set which corresponds to some translated regions there. So we have developed an algorithm that is able to very efficiently actually to very efficiently find the frame shifting sites and to find generally coding sequences in these situations when you have abundant frame shifting. So and it is very specific and very precise. So it's so it has like TPR of approximately 70% on FPR of 0. And so we study the evolutionary dynamics. We see that most of the frame shifting sites arise by the insertions of thymines, which yield these stop codons, and they're lost by the corresponding deletions of thymines, and so we kind of regarded this situation as it accounted for like 70% of generally frame shifting sites gains and losses. And generally, we see more frame shifting site gains. But if we look at the probability of gains and losses, we see more probability of a frame shift site loss. This may happen if we have a non-equilibrium situation here. So the sites are accumulating. So we have a differential equation here which we solve, and we get like the projections of the frame shift site numbers over time. We see that the frame shift site numbers are expected to increase like several folds in the course of a long time. So it's going to take time equivalent to 3 or 4 synonymous substitutions per site. So yeah, and yeah, and that corresponds to like billions of years. Then we have calculated these selection coefficients, and we have found that essentially these frame shifting sites are mildly deleterious. So if we multiply the selection coefficient by the effective population size, then we get approximately 2, which corresponds to sort of an in-between situations where both selection and drift influence this accumulation of frameshift sites, and then we have assessed that basically if the numbers of frameshift size at equilibrium will yield a lag load which will be substantial, and we find it to be not substantial. So essentially this kind of novel feature of the genetic code will propagate all across the Euplotes genomes, and it will become their inherent feature. And considering the emergence of such a situation, we put forward a hypothesis that once a translation apparatus gains this ability to frame shift, then the corresponding insertions of thymines start accumulating, thus basically entrenching this situation, entrenching the translation apparatus, and so they cannot kind of get back to this situation when they do not have the frame shifts. They can only accumulate them, even though it is a mildly deleterious mutation.